Hello again biologists, Mr. Kinnear here with another video lesson for higher biology. We're on Unit 1, DNA and the genome, and today we're going to look at structure and function of RNA. So this actually falls within key area 1.3, gene expression. So in future video lessons I'm going to talk about that process of transcription and translation, but today I'm just going to give you a bit of a heads up on RNA because that's very important in those two processes. So gene expression is what makes all of these individual people the individuals that they are. And you and I together as well. We share about 99.5% of our genetic material, the genetic code that makes us who we are. We also have about 95% of the genetic material the same as orangutans and chimpanzees, our closest ape relatives. So a very small amount of our genetics makes us different. And that is about gene expression, which is about the proteins that we produce within our cells. And that's what we're going to look at over the coming video lessons. Gene expression and making us who we are. You should be able to recognize these two cells from National 5 biology. On the left we have a muscle cell, on the right we have a nerve cell. If both of those cells came from you, you should know now that both of them will contain exactly the same DNA. When it's in the process of mitosis, each mother cell produces two identical cells. And again, we looked at the DNA replication of how that happens. Now, what's the thing that makes them different? Well, it's the gene expression. It's which one of those genes that it contains that are switched on and which ones are switched off. And effectively, it's like a recipe book. So each of your cells contains the recipe to make you. But a muscle cell does not need to use all of those recipes. It just wants the ones that are important for being a muscle cell. And equally the nerve cell. It has the options for all of the recipes, but it's only interested in the recipes that make it a nerve cell. So that's gene expression. It's about taking the right recipes out of the recipe book and making the right proteins that is required to do its job. So we've got three things to learn today. How does phenotype arise? What influences phenotype, and the structures and functions of RNA. You should remember from National 5 the word genotype. That reflects the combination of alleles that you receive. Obviously 50% from mum, 50% from dad. That is then reflected within your base sequence. And that is not something that we could look at and see directly. You could obviously go and get some genetic screening and they could then give you your base sequence or your alleles or your genotype. But the most common thing is that we see how that is reflected in the amino acids and then the proteins that are produced. And that could be hair colour or eye colour. Because that moves us on from genotype to phenotype. So we do have the base sequences making up our genotype. That then determines the proteins that are produced by gene expression, which then determines the phenotype or physical characteristics of the organism. So a cell's phenotype is its physical and chemical state, which is determined by the proteins that are synthesized when the genes are expressed, which is called protein synthesis. And here's a rough diagram of protein synthesis. As we remember from National 5, there are two stages to it, and we're going to look at those two stages in a lot more detail in the coming video lessons. But we should also know that phenotype can be affected by environmental factors inside and outside of the cell. So extracellular, outside, environmental factors including drugs, chemicals, temperature and light. We call these outside factors epigenetic modification or epigenetic modulation, as you can see from that diagram. It's where the DNA itself is not changed, but how it's expressed is due to other outside factors. And it's how you can have identical twins who end up growing up looking different. Although they may have the same genetic material, how it's expressed is modulated by life experiences and all the bits and pieces you can see on that diagram. We did look at RNA at National 5 level, so you should be familiar with how it's similar to DNA and how it's different. But we'll look at that again just to make sure you're 100% comfortable with it. Things that you should remember from National 5, RNA is a single-stranded molecule, whereas DNA is a double-stranded molecule. RNA is a lot shorter as it carries the code for one gene, whereas DNA carries all of your genetic material and therefore a lot longer molecule. You'll also remember the bases, so cytosine, guanine, adenine, thymine on our DNA, whereas we have cytosine, guanine, adenine and uracil. And again, the complementary base pairings, so again U takes over from T on RNA. And although this may have been discussed at National 5 level, it wasn't part of the course learning, but DNA is obviously known as deoxyribose nucleic acid, RNA ribose nucleic acid. And the difference being that deoxyribose sugar in DNA is missing an oxygen, whereas ribose has the oxygen present. So the deoxyribose, our DNA, is missing the oxygen. So that's another part and parcel of our differences in the structure. Our ribose sugar has the oxygen and our deoxyribose does not. Here's a quick summary of that key information regarding the structural differences between DNA and RNA. DNA, double-stranded, contains deoxyribose sugar and contains thymine and a very long molecule. Our RNA, however, being single-stranded, contains ribose sugar, has the base uracil and is a lot shorter. And we're now going to talk about the types and functions of RNA molecules. At National 5 last year, we would have talked about messenger RNA or mRNA. It carries a copy of the DNA code from the nucleus to the ribosome. It's involved in both transcription and translation stages of protein synthesis. And a key bit of information, 3 bases is called a codon, and each of those codon codes for a specific amino acid. So we now need to be specific when we're talking about RNA and use messenger or mRNA, because I'm going to introduce you to the second type, which is transfer RNA or tRNA. Its role is to carry a specific amino acid to the ribosome. It's involved in the translation stage of protein synthesis. 3 exposed bases is called the anti-codon, and as its name suggests, the anti-codon is complementary to the codon on the mRNA molecule. And we know from last year that protein synthesis happens at the ribosome, and it contains ribosomal or rRNA. That's bound to structural proteins to form the ribosome, where the translation stage of protein synthesis occurs. So I'll take you through some more detail regarding the tRNA molecule, which you can see in this diagram. And it's a single-stranded molecule, but it is folded in upon itself, with hydrogen bonds forming in 4 key areas to produce this shape. So we've got hydrogen bonds between 2 different parts of the same strand. It also contains a triplet anti-codon down here at the bottom, which, as we said before, is complementary to the codon on the mRNA molecule. It also contains an attachment site for a specific amino acid. And each tRNA molecule binds with a different amino acid depending on the anticodon it contains. I often think about transfer RNA as a taxi. Its job is to taxi one amino acid from the cytoplasm to the ribosome, leave it there because of the code or the codon that is there present on the mRNA molecule. And that complementary system allows it to drop it off at the right place and at the right time for our protein synthesis. We'll talk about that in more detail when we get to translation. At higher, we need to start to decipher the genetic code and understanding the relationship between DNA and its complementary RNA molecules. So our genetic code starts with DNA, and 3 bases together on DNA would be referred to as a triplet. As you can see, I've got 3 triplets here: ATG, CCG, TAT. Now each of those triplets would code for an amino acid to be joined together at the ribosome to form that polypeptide. But it has to go through a couple of steps first. The first of those steps being transcribed into mRNA. And on mRNA, 3 bases is referred to as a codon, each of those codons being complementary to the triplet. So I've got UAC, GGC, AUA. So I'm putting a U where I see an A where I see a T, a C where I see a G, and a G within a CSC, as you can see from those triplets and codons together. The next step is for my tRNA molecules to bring over the specific amino acids to be joined together. And on tRNA, I've got an anti-codon. An anti-codon is 3 bases, which is complementary to the codon. So the 3 anti-codons would be AUG, CCG, UAU, complementary to my 3 codons on my mRNA molecule. You might start to see a relationship between the triplets on DNA and the anti-codons on tRNA. They're pretty much the same with the exception of U's on my tRNA, where I have T's on my triplets. So these are things you might be asked to do within exam questions, is convert DNA into tRNA or tRNA back into DNA. You normally have to do a couple of steps there within higher level questions, remembering that complementary rule and substituting the different letters between DNA and RNA. So that's us for another video lesson. Today we've looked at phenotype, the physical and chemical state, which is determined by genetics or your genotype, and other factors such as epigenetics, so external factors. We've also looked at there being 3 different RNA molecules, which is new information, mRNA, which you've come across before, but also now tRNA and rRNA, and they all have individual functions within the protein synthesis process. And we've also talked about anti-codons on tRNA, which are complementary to codons on mRNA, which are then complementary to triplets on DNA, and how you need to understand that relationship, more of which we'll talk about in coming lessons on protein synthesis. So as usual, if you have any problems, questions or queries, get in touch and I'll speak to you again soon. Bye.